So now we're going to move on and talk about purification steps. Where do we start? Well, the first place we start is we got to break open the cells. Now, there's some exceptions to this, right? Some of the first proteins to be studied, nobody had to break open any cells. That's because they just studied proteins in your blood, right? We could just take blood from you, spin the cells down, take what's left that's not clotted called plasma. We'll talk about that shortly. But if we want to do this, we have to find a way to break open the cells. And there are a bunch of techniques we can use. We can grind the cells up, right? We can either grind them up by themselves, we can throw in some sand, we can do anything we want to grind the cells up. We can freeze and thaw them. Freeze, thaw, freeze, thaw, freeze, thaw. You break the membranes. That's one way to do it. You can use a detergent, something that disrupts the cell membrane. You can use sound waves to shear the protein apart. My favorite, and the one that's probably the best of all of these, is rapid pressure changes. I should have written the word rapid. Rapid pressure changes. Okay, an example of this is something called a French press. I know. A French press is that thing you put the coffee in the morning and you pour the hot water. No, this is a different French press. Why does rapid pressure work? Well, imagine that I take some cells and I put them in a high pressure bomb and I turn the pressure up to 500 atmospheres. Okay? What happens? Well, the cells equilibrate, right? The pressure outside is very high initially, but the cells rapidly equilibrate. And now the inside of the cell is at 500 atmospheres. The outside cell is at 500 atmospheres. Now what do? I release the pressure outside. Now what happens? Suddenly inside the cell is 500 atmospheres. Outside the cell it's 1 atmosphere. Kaboom! The cells explode. Works really well. Very, very gentle. So now I've got some cell goo at the end of this, right? And now I want to figure out what I'm going to do with this. So I got to go one step further after I've broken the cells open, and I got to somehow separate this into the next steps. So to do that, the next step I'm going to do is I'm going to separate them by centrifugation. Okay? So I'm going to take my cell goo, and I'm going to spin it at, say, low speed, which for this class, let's just say 500 times gravity, okay, for 10 minutes. And I'm going to end up then, in my tube, I'm going to end up with a pellet, and I'm going to end up here with some supernatant. And when I do this, in fact, the pellet at this speed contains nuclei. So at this point, I'm going to take the supernatant, and I'm going to spin it again. This time, I'm going to spin it at medium speed. And medium speed here is about 10,000 times g. And I'm going to spin this for about 20 minutes. And I'm going to end up again with a supernatant and a pellet. And the pellet that I end up with this time contains mitochondria. Now, at any point in time, I can stop, right? I can say, oh, I'm really interested in what's going on in the nuclei, and I can work with them. Or I can say, oh, I'm interested in the mitochondria. But if not, I still have a bunch of stuff here. So I could take the supernatant again, and I could spin it again, this time at high speed. And by high speed, I mean 100,000 times the force of gravity for, say, 2 hours. And now I'm going to end up with a pellet and a supernatant. And my pellet, when I do this, will be primarily microsomes. And microsomes are nothing more than ribosomes and membranes. Basically rough ER. And I'll end up with a supernatant here. And this supernatant that we're left with here, we're going to call cytosol. That's the sap inside cells. Okay? So the supernatant at this point equals cytosol. Okay? Now, let's just say my protein is sitting here in the cytosol. So now I need to do the next step in order to work to get that out. So to do this, now the next thing I have to do is I'm going to make use, I guess I should write this over here. How am I going to get my protein out of this cytosol? Well, I'm going to now use differences in solubility, charge, size, or affinity.